Good morning, everyone. Today, I'm going to share with you the topic medicinal chemistry. Initially, medicinal chemistry is a branch of science that combines chemistry, biology, and pharmacy to discover, design, develop, and optimize drugs for the treatment of diseases. It includes these scientific disciplines at the intersection of chemistry and pharmacy involved with designing and developing pharmaceutical drugs. Medicinal chemistry involves the identification, synthesis, and development of new chemical entities suitable for therapeutic use. It also includes the study of existing drugs, their biological properties, and their QSAR. It's denote qualitative structure activity relationship. Move on to the next slide. Exploring the dimensions of medicinal chemistry. Medicinal chemistry plays a vital role in drug discovery and development. It involves understanding disease, identification drug targets, designing molecules, testing their activity, and improving their therapeutic effects. Medicinal chemistry is a branch of science. It is disciplines with the intersection of chemistry and pharmacy. It focuses on identification, synthesis, and development of chemical entities. Its main goal is to produce safe and effective medicine. Exploring the dimension of medicinal chemistry, it includes the chemistry, biology, pharmacology, drug discovery, healthcare impact. The next slide. Components of medicinal chemistry. The major components include drug discovery and design, then QSAR, structural activity relationship, pharmacology, design, drug synthesis, drug metabolism, and pharmacokinetics. These components help scientists understand how drugs work and how they can be improved, how drugs exert their effect at the molecular level, the relationship between a compound structure and its activity, the effects of drug on biological system, the methods used to manufacture drugs, the process of creating new pharmaceutical compounds. The next slide. Stages in medicinal chemistry. Medicinal chemistry covers the following stages. Number one, identification of lead molecule. Number two, optimization of lead structure. Number three, development of synthetic route for large scale production. These stages ensure the development of effective and safe medication. The optimization of lead structure include focuses on enhancing the drug's effectiveness and safety. Number two, development of synthetic route involves creating effective methods for large scale production. Number three, identification of lead molecule. This stage involves discovering new drug candidates from various sources. Lead molecule. A lead molecule is a compound that shows promising biological activity against a specific disease target. It serves as the starting point for drug development. Scientists further optimize the lead molecule to improve efficacy and safety. Lead molecules are typically identified through high throughput screening, computational modeling, or medical chemistry efforts. It serves as the starting point for further research and development to optimize its property, include efficacy, safety, and drug-like characteristics. High throughput screening. High throughput screening is a technique used to test thousands of compounds rapidly. It helps identify potential lead molecules that can be developed in drugs. The next slide. Computational modeling. Computational modeling used computer-based techniques to predict how drug molecules interact with biological targets. It reduces time and cost in drug discovery. Then, optimization of lead structure. Lead optimization involves modifying the chemical structure of a lead compound to improve potency, selectivity, safety, and pharmacological properties. Then, optimization of drug candidates. This stage focuses on enhancing drug effectiveness while minimizing toxicity and side effects. Scientists make structural modification based on SAR studies. Then, evaluation of medicinal chemistry. Medicinal chemistry has evolved from the use of natural remedies to modern computer aided drug design and biotechnology based therapeutics. Then, the historical development. Early medicines were derived from plants and natural products. Significantly, milestone includes isolation of morphine from opium, discovery of aspirin, discovery of penicillin by Alexander Fleming in 1928, development of synthetic and targeted therapies, solubility and drug properties slides, pharmaceutical revolution of 20th century. Alexander Fleming discovered penicillin in 1928. Synthetic antibacterial like sulfur drugs were also developed. Then Paul Sandwich coined the term magic bullet and developed salvarsan, the first effective drug for syphilis. Aspirin, first widely produced synthetic drug, barbiturates and benzodiazepines were synthesized for neurological and psychiatric conditions. Development of drugs to cancer treatment, such as methotrexate and cisplatin, represented milestone in medicinal chemistry. Modern era, 21st century and beyond. Development of drugs like imatinib for cancer and monoclonal antibodies for autoimmune disease and cancer. Automation and computational chemistry allow rapid testing of thousands of compounds. Focuses shifted to biological derived drugs like insulin analog, monoclonal antibodies, and gene therapies. Innovation include liposomes, nanoparticle, and mRNA delivery technique, example COVID-19 vaccines. AI derived drug discovery accelerated the identification of novel compounds and reduces the cost of research and development. Physiochemical properties of the drug. Initially, solubility, hydrogen bonding, ionization, surface activity, chelation, partition coefficient, dissociation constant, molar refraction, protein binding, bioisomerism. The solubility. Solubility is an important property affecting the drug absorption and bioavailability. Factors including solubility include hydrogen bonding, dipole-dipole interactions, van der Waals force, chemical modification of drug molecules. Hydrogen bonding. Hydrogen bonding is a type of weak intermolecular force that focuses on hydrogen atom that is covalently bonded to highly electronegative atom. Form an attraction with another electronegative atom in a different molecule or part of same molecule. Dipole-dipole interaction. Dipole-dipole interactions play a significant role in solubility, particularly when it comes to the dissolution of polar substance in polar solvent. Follows the principle of like dissolves like. Polar substance tend to dissolve well in polar solvent, where the nonpolar substance do not permanently dipole and can't from dipole-dipole interaction with polar solvents. Van der Waals force. Van der Waals force or a group of weak intermolecular force that plays an important role in the solubility of both polar and nonpolar substance. Methods to improve solubility. Number one, structural modification. Number two, use of co-solvent. Number three, employing surfactant. Number four, complexation. Structural modification. Introduce polar group. Reduce molecular size. Number three, create salt form. Identify polar soluble compounds. Use water alcohol mixture. Combine solvent with different polarities. Surfactant, miscellaneous formation, encapsulation of hydrophobic molecule, pharmaceutical industry, Twine 80 and SLS. Then finally, conclusion. In conclusion, the medicinal chemistry is a crucial field in modern healthcare. It bridges chemistry and biology to discover and develop safer and more effective drugs. Advanced in medicinal chemistry continue to improve disease treatment and patient outcome. Remaining, we have seen in part 2. Thank you.